and welcome to our Hearing Health Foundation research webinar. I'm Dr. Anil Lawani, and I appreciate you joining us today. Please note that this event has a live captioner. You can enable closed captions by clicking the CC button in the toolbar at the bottom of your screen. Now, if you need any other assistance using Zoom, please follow the link to the technical guide shared in the chat. Please note that unlike our other webinars, this webinar is not being recorded, and we will provide a brief bibliography to all those who registered, but we will not be recording this webinar. Now, by way of introduction, my name is Dr. Anil Lawani, and I'm a professor and vice chairman for research in the Department of Otolaryngology, Head and Neck Surgery, as well as Associate Dean for Student Research at Columbia University Vangelos College of Physicians and Surgeons in New York. And thus the big buildings behind me. I'm also a board member at Hearing Health Foundation and the head of the Hearing Health Foundation's Council of Scientific Trustees. Now, the scientific trustees oversee the Emerging Research Grants Program, affectionately known as ERG. Now, ERG is a competitive program that awards funds to researchers conducting cutting-edge hearing and balance research. Now, these grants supported many leaders in our field to become successful scientists, including our speaker today. Now, our presenter today is Susan Shore, PhD. Dr. Shore led groundbreaking research on how the brain processes bisensory information and how these processes can be harnessed for personalized stimulation to treat tinnitus. In this webinar, she will describe the basic science and the research she has conducted over the past 2 decades that has led to promising therapy for tinnitus. Dr. Shore received her PhD in physiology at the Kresge Hearing Research Laboratory of the South, located in New Orleans at Louisiana State University Medical Center, and her postdoctoral fellowship included in the University of Pittsburgh and the University of Michigan Medical School. An emerging research grant scientist from 1987 to 1991, 1987 and 1991 to 1995, Dr. Shore is a professor emeriti in Michigan's Medicine Department of Otolaryngology and the University of Michigan's Department of Physiology and Biomedical Engineering. So she is a member of a lot of different departments because her contributions are so wide. She's also a co-founder and the chief scientific officer of Oracle Incorporated. Now, the ERG program that provides seed money to scientists just starting out in their field of research is only possible through the generosity of supporters like you. If you'd like to support our work on hearing loss, tinnitus, and related conditions, you can do so today at hhf.org/donate. hhf.org/donate. Now we'll move to Dr. Shore's presentation. Please ask questions to the QA box linked at the bottom of the screen that we'll try to answer following the presentation. We hope that you understand that Dr. Shore cannot answer any questions about or discuss the FDA approval process or timelines regarding the availability of the device she has developed with Oracle. She's also unable to discuss other therapies or treatments, as this would be a conflict of interest. Thank you so much for understanding. And without further ado, Dr. Shore.
Thank you, Anil. Can you see that okay?
Yes, we can.
And thank you so much for the invitation. It's an honor to present at this seminar. And I will do my best to make this understandable to a wide audience and not just to scientists. So be patient. Okay, so first I need to provide these disclosures, which Dr. Lalwani already has done partly, but to acknowledge other sources of funding, NIH for the past 30 years or so, as well as the American Tinnitus Association and local funding from the University of Michigan. So I'd like to just start out by presenting some concepts that you're going to need to understand for this talk. First, what is tinnitus? If I had an audience and I could ask you questions, I'm sure I'd get a number of answers that are containing different kinds of sounds. But the definition that I'm looking at that's important here is that it's the perception of sound in the absence of an auditory stimulus. And because of that, when we're studying tinnitus in an animal model or even in a human model, we need to be aware of the spontaneous activity, because this is the activity that occurs in the absence of sound. So you're going to hear me today talk a lot about the spontaneous activity in the neural structures that I'm going to show you are responsible for generating the tinnitus. These studies went over a couple of decades, and we had to first ask, what does the normal system look like? Because you can't really study a pathological system without knowing what the normal system looks like in the first place. And then you have to look and see what the system looks like after noise damage, which was our model for tinnitus, as the most common factor associated with tinnitus is noise exposure. But then that's not good enough, because you also have to look at what happens after tinnitus induction, because noise exposure doesn't necessarily mean that there's going to be tinnitus there. Not all people or animals get tinnitus after noise damage. And those without tinnitus can tell us something about resilience or protection. When we're doing basic science studies, we have to know whether the animal has tinnitus. And because of that, we have to have a behavioral and a physiological model that's going to tell us that an animal has tinnitus or doesn't have tinnitus. So today, our focus is going to be on the cochlear nucleus, which is seen here as part of the brainstem, and it sits here between the medulla and the pons. And you can see that the cochlear nucleus is the only structure that receives direct input from the cochlea, from the auditory nerve fibers. And because of that, the cochlear nucleus is going to be directly affected by whatever happens in the cochlea. So if there's cochlear damage, say from noise exposure, the neurons in the cochlear nucleus are going to be directly affected. Now, of course, even though I'm going to show you that tinnitus is generated, the tinnitus signal is generated in the cochlear nucleus, in order for a person to perceive that that signal is tinnitus, this signal is going to leave the cochlear nucleus and go to several central structures, including the inferior colliculus, the thalamus, and eventually the primary auditory cortex, where we're going to perceive that signal as tinnitus. So now let's look in a little bit more detail about the actual circuit. And today we're going to focus on the dorsal cochlear nucleus. There are two divisions, the ventral cochlear nucleus, which is important as well, but not for today's talk. And so this is a schematic of some of the elements in the dorsal cochlear nucleus. The ones important for this talk, in particular, are these cells here. They're called fusiform cells, abbreviated FC. And these are the principal output cells of the dorsal cochlear nucleus. And they send their outputs to the inferior colliculus and to the thalamus, the medial geniculate body. You can see that these cells are very special in that they have two sets of dendrites. Those on the top are called the apical dendrites, and those on the bottom are called the basal dendrites. The basal dendrites are the ones that receive the information directly from the cochlea in the form of auditory nerve fiber inputs. And the parallel fibers receive inputs from other sensory systems, including the somatosensory system. So you can see here that there are a number of different somatosensory inputs and other kind of multimodal inputs that send their inputs into the dorsal cochlear nucleus by way of these parallel fibers. So you're going to hear me talk a lot about parallel fibers and auditory nerve fibers. And the thing to remember here is that these parallel fibers contain the somatosensory inputs that come from various different places in the body. And I'm going to show you that these synapses between the parallel fibers and the apical dendrites of these fusiform cells is where the long-term plasticity is that is going to drive our studies on tinnitus. So moving on here, now what I'd like to show you is some early studies that showed that these parallel fiber synapses, shown here again, are plastic. And these were studies done by this group in 2003, where they showed that depolarizing the postsynaptic cell, in this case, is a fusiform cell, and the cell can be depolarized in various different ways. So these cells were depolarized here with electrical stimulation, and then the parallel fibers were stimulated. And with repeated stimulation of the parallel fibers, they were able to generate long-term plasticity in these cells called long-term potentiation, or LTP. In addition, they were able to generate long-term depression in the fusiform cells by low-frequency stimulation. So they depolarized the cell while stimulating the parallel fibers at a low frequency. And they were able to generate this long-term depression. So in the case of long-term potentiation, the cell is being made more excitable, and this lasts for a long time. Whereas in the case of long-term depression, the cell is being made less excitable, and this also lasts for a long time. Now, this group went on to further study the circuit, and they did the following experiment where they stimulated the parallel fibers, evoking EPSPs in the parallel fibers, followed by spikes that they got by stimulating the fusiform cells. And if they stimulated in this particular order with the EPSPs before the spikes, the EPSPs before the spikes repeatedly, they ended up getting long-term potentiation. But interestingly, when they reversed the order and they stimulated the spikes before the EPSPs, they got long-term depression. So in this cell, they were able to either generate long-term potentiation, LTP, or long-term depression, depending on the stimulus order and the interval between the two stimuli. So for example, here, with an interval of 10 milliseconds, you get the most long-term potentiation, and you get the most long-term depression at an interval of about 7 milliseconds. So I'd like you to keep this in your mind. We're going to come back to this a few times, but before we do, I'm going to move on to some other studies. And here's a summary of what I'm going to talk to you about. So I'm going to show you that the fusiform cells in the dorsal cochlear nucleus encode tinnitus in their spontaneous firing rates and in their spontaneous synchrony. And then I'm going to show you in vivo, in the whole live animal, that these fusiform cell synapses are plastic, that these are the synapses that come from the somatosensory system. Then I'll show you that this plasticity is altered in animals with tinnitus, and I'll show you how we figured out that we can reverse this plasticity to treat tinnitus. And finally, I'm going to show you how this is applied in human patients. So just to give you some idea of how we study that somatosensory fibers innervate the cochlear nucleus, we put what is called a retrograde tracer. We inject this into the live animal cochlear nucleus, and these are guinea pigs. And then after the animal is sacrificed, we section the brains, and we look to see where there are labeled cells indicating that these areas in the brain send projections into the cochlear nucleus. And what we and others found is that the spinal trigeminal nucleus and other places as well that are not somatosensory necessarily, but the somatosensory inputs come from the spinal trigeminal nucleus and also from gracilis and cuneate nuclei, which are in the spinal cord. And these are all conveying somatosensory information into the cochlear nucleus. Now, we also showed importantly that's necessary to understand these studies that these somatosensory inputs, which terminate in the cochlear nucleus, and they label with vesicular glutamate transporters because they're glutamatergic. So here we're showing some labeling of presumed somatosensory inputs that co-label with vGlut2. And in the top, we're showing auditory inputs, auditory nerve inputs that co-label with vGlut1. But because these label with different markers, we're able to trace these synapses and see what happens to them after we induce cochlear damage. And so we did that. In this first study, we deafened the animal. So it was a very severe cochlear damage. And you can see here, as expected, that the inputs from the auditory nerve are gone. So so as I said to you, that the cochlear nucleus will reflect what happens in the cochlea. So in this case, the auditory nerve fibers are gone because we've produced severe cochlear damage. But what happens to the somatosensory inputs? Well, you can see here that the somatosensory inputs increase. So where we get a decrease in auditory nerve inputs, we get a compensatory increase in somatosensory inputs into the cochlear nucleus. This is called homeostatic plasticity. And this also occurs after very mild cochlear damage, as well as this severe case that I've shown you here. So now, having that in your mind, I'm going to remind you of this circuit diagram that I showed you earlier. And I'm also going to remind you that the somatosensory inputs are going to be coming in through these parallel fibers, whereas the auditory nerve inputs are going to be coming in through these, the basal dendrites of these fusiform cells. So to remind you what the studies done by other groups have shown in vitro, in that you can get LTP or LTD depending on the stimulus order and the interval between the stimuli, in this case, parallel fiber and spikes in the cell. So that's a way of generating what we call spike timing dependent plasticity. Spike timing because of the timing of activating these spikes to the stimulation of the parallel fibers and spikes evoked in the fusiform cell. So now the next step in this research has to be, well, how do we generate spike timing dependent plasticity, or how do we show it in the live animal? And so that was what the next set of studies is going to show you. And so here again, we have our fusiform cell circuit. And what I'm going to show you is what happens when we stimulate these apical dendrites by stimulating the somatosensory nuclei. And to do this, we first stimulated the trigeminal nucleus with deep brain stimulation. And then we stimulated the auditory system with a specific timing, either before or after the somatosensory stimulation. So in this case, we're going to be generating LTD or LTP, and you'll be able to see which. So first we have somatosensory stimulation followed by auditory stimulation. And then I'm going to be plotting on this graph what happened. And so in this case, we got an increase in spike rate, which is how we're measuring our LTP. And this is long-term plasticity. So this change happened for a long time. And now I'm going to show you what happens when we do auditory followed by somatosensory stimulation. And in this case, we got long-term depression. And so we're able to replicate the spike timing dependent rules, or the spike timing dependent graphs that demonstrate how the circuit responds to these stimuli. And so now we know we can generate and measure spike timing dependent plasticity in vivo in our model. So we want to know now if this is the way the normal system behaves, and this is how the plasticity is represented in the normal system, what's going to happen in an animal that has tinnitus? So in order to first do that, we're going to first have to do an experiment where we show that we have tinnitus in animals. And so for this reason, we used this particular test called the gap detection test, where we got a baseline of gap detection, and the animals were divided into two groups, an unexposed sham control group and a noise exposed group. The noise exposed group received 90 decibels of noise for about an hour and a half. And then we followed these animals over 8 weeks. And these animals with the gap detection technique demonstrating whether they had tinnitus or not, 40% of them did not have tinnitus and 60% of them did have tinnitus. So what happens to fusiform cell spontaneous activity now in animals with tinnitus? And so in order to do that, we put an electrode in the dorsal cochlear nucleus, a multi-channel electrode, and we record along the tonotopic axis of the dorsal cochlear nucleus. And here we're now plotting the spontaneous rate. This is showing the noise exposure paradigm. So this is centered 8 kilohertz noise. And now we're going to show across the best frequencies along this tonotopic axis what the spontaneous rates look like 8 weeks after post-exposure or post-sham exposure. And so for the control sham animals, this is the baseline of normal spontaneous activity across this axis. Now, the animals that got noise exposure but that did not develop tinnitus show pretty much the same profile of spontaneous activity as the normal animals. And this is what happens to the animals that get tinnitus. So not only do we have a behavioral estimate of the tinnitus, but now we also have a physiological estimate of the tinnitus in the increased spontaneous rates that we see above the noise exposure spectrum. Now we also want to look at, and of course what I am showing here is that many groups have shown this increased spontaneous activity in the dorsal cochlear nucleus. What we went on to show was that not only spontaneous activity is increased, but also synchrony between the neurons is increased. And this is important because if you think about tinnitus as something that you perceive, it can be considered like an object. And for object perception, synchrony between neurons is very important. So again, here we show the normal animals and the animals without tinnitus compared to the animals with tinnitus. So again, now we have physiological correlates of tinnitus being increased spontaneous rates and increased synchrony, increased spontaneous synchrony between fusiform cells. Okay, so and now we know we have the basis for showing that we can show spontaneous activity in the fusiform cells that is spike timing dependent. And if we're going to now think it forward about how we could develop something that could apply to tinnitus, we need to be able to use a more non-invasive method to do the stimulation and to demonstrate the circuit, how the circuit responds to these sounds. And so instead of doing deep brain stimulation to produce our LTD and LTP, as we've shown here, just doing this again. We're doing the same thing now, and we're showing, sorry, this is not the non-invasive technique yet. This is just showing now what happens to spike timing dependent plasticity in animals that have tinnitus. And remarkably, they show the complete opposite pattern of what the normal animals show. So stimuli that in the normal guinea pig would cause LTP are now causing LTD in animals with tinnitus, and vice versa. So stimuli that produce LTD in the normal animal produce LTP in the tinnitus animal. And this demonstration demonstrates a very important point in tinnitus research, is that you can't make assumptions about what the tinnitus circuit looks like by looking at a normal circuit, because the circuit changes after both noise exposure and tinnitus induction. So you can see here, not only is this spike timing dependent curve reversed, but it's also shifted upwards. And so you can see there's less long-term depression and there's more long-term potentiation, indicative of a more excitable circuit. Okay, so now we're getting to how we do this same paradigm, but using less invasive stimuli. And so we developed an animal model where we could stimulate the somatosensory system by stimulating the neck. This is stimulating basically the cervical nerves. And we know that we were stimulating these because we measured responses in the fusiform cells to these stimuli. And we stimulated the animals again with sound. So we're doing bisensory stimulation again. And we're going to regenerate our spike timing dependent curve. So this is the control animal. And this is what the animals with tinnitus look like. So we're showing again with this non-invasive stimulation that we can reverse the spike timing dependent plasticity, that the tinnitus circuit has reversed the timing. And interestingly enough, the tinnitus animals and the no tinnitus animals do not show the same profile. So that leads me to make my second point, is that you can't just do a noise exposure experiment and assume that that's going to be the effect on tinnitus, because animals that don't develop tinnitus do not show the same profile that animals that do develop tinnitus. And so here you can see that the animals that develop tinnitus have a more excitable circuit. They've got more long-term potentiation and less long-term depression than both the controls and the no tinnitus animals. So in some way, these no tinnitus animals are showing resiliency, and they're not developing this tinnitus circuit. So thinking then about how is this going to help us lead to a treatment for tinnitus, we reasoned that there are intervals, bisensory intervals that cause long-term depression. So what happens if we take this particular stimulus and we repeat it to the animal and present it every day for a half an hour? Is that going to make the circuit change? We thought, yes, it is going to make the circuit change. So in fact, that is what we did. We got another group of animals. We had an unexposed control and a noise exposure group. Some of them developed no tinnitus and some of them developed tinnitus. And then those that developed tinnitus got several types of treatments. The bimodal or bisensory treatment, which was the treatment we'd shown in the previous animals to produce long-term depression. And then we had a somatosensory alone, which we know will cause some long-term potentiation by itself. And unimodal auditory stimulation, which we know will produce no plasticity because the inputs to the basal dendrites are not plastic. And so we did this transdermal electrical stimulation combined with an 8 kilohertz tone for 20 minutes a day for 25 days. And in fact, we were able to reduce the animal's tinnitus. So this is just showing the different groups. And this is showing a tinnitus index, which is the amount of tinnitus. So first of all, the ones that showed no effect with the sham animals, so their stimulation showed no effect on them. The ones that received somatosensory stimulation alone showed an increase in tinnitus by quite a lot. And a couple of animals also showed some decrease. So it was a very variable response. And then the ones that received the tinnitus treatment at 8 kilohertz all showed a decrease in their tinnitus. So repeating the long-term depression for half an hour a day for 1 month reduced all of the animals' tinnitus. And if we of course recorded from the animals' brains as well, and we can show a very nice correlation between the tinnitus index and in this case, which is the amount of synchrony. So again, here are the animals that received the treatment. Here's the somatosensory showing the highest synchrony. And by the way, also spontaneous activity correlating with the tinnitus index. So now we want to see what does this particular treatment do in humans. And so for a pilot study, we recruited 20 adults with chronic tinnitus that had somatosensory involvement. We picked this group because the somatosensory system is heavily involved in this treatment. these patients had normal to mild hearing loss, and we had several exclusion criteria, including not participating in any tinnitus studies within 6 months of the study. The non-treated ear and the treated ear showed equal amounts of cochlear damage, just in the higher frequencies here. So the treatment was based on the animal studies. And we had a bisensory auditory somatosensory treatment, where the auditory stimulus was the same as the tinnitus spectrum, which we measured using an interactive computer program. And this was important because if you noticed in the previous slide, the animals that got a tone that was different from their tinnitus did not show as much improvement as those in which the sound was the same as their tinnitus. So you can think of the auditory stimulus as activating those pathological neurons at the tinnitus frequency. The transdermal electrical stimulus was on the face of the neck, as shown here, and it was placed just above threshold so that the patients could hardly perceive it, and after a while, they couldn't perceive it anymore. The bisensory interval was derived from the preclinical studies. This is the clinical trial design. So we had a baseline, and then subjects were randomized into either an active first or a sham first. And so we had a sham treatment, which was the electrical stimulation alone. And the active treatment was the bisensory treatment. They received this for 4 weeks. And then there was a 4-week washout period, after which they switched to the other treatment. So shams got the active and the active got the sham. And then after that, there was another washout period. So these are the results. We measured change in loudness, which we also used the interactive computer program for. And you can see here that over the 4-week period, we see a progressive decrease in loudness of those that received the active bisensory treatment. And at the end of this period, it came back up to baseline. And then when the group switched, you saw the identical result. So this progressive decrease in loudness over the 4 weeks. The auditory alone stimulus did not produce any, I think I said somatosensory alone before, I meant auditory alone, did not produce any results, as we had not anticipated, as the auditory alone does not produce this plasticity that we see with the somatosensory stimulation combined with the auditory. We also measured change in tinnitus functional index, a questionnaire looking at the life impact. And here again, we see a progressive decrease in the tinnitus functional index, which levels off and interestingly continues in the washout period, but eventually comes back to a baseline. And the same thing with the auditory stimulus, it has very little effect. So there were significantly greater decrements in TFI and in loudness in the active group than in the sham group. And so given these very encouraging results, we went on to a larger study where we recruited 99 people, and we followed the same plan as we did with the first study, in that it was a double-blinded sham crossover study. The only difference was that this length of the study was 6 weeks instead of 4 weeks. That's what I just said. And it was the same stimuli that we used in the previous study. The interval was from the same as the animal study and from the preclinical the preclinical and the phase 1 trial. So this is now just comparing the two groups, and you can see the only difference is the 6 weeks. And these are the results. Here we're showing the change in the TFI score. And you can see that we're getting a much better result than we got in the first study with the same trend. So we're seeing a progressive decrease in the TFI And this reaches clinical significance, which is 13-point reduction by the end of the 6 weeks. And remarkably, this continues to decrease in the washout period where there's no stimulation whatsoever. We did not expect this. We expected that with an additional length of the washout period, we would get a return back to baseline. But because we didn't get a return back to baseline, we couldn't use the second half of the study, which of course is a great result. And the reason you can't use the second half of the study, the crossover, is because you're still seeing a treatment effect of the active treatment. So any other treatment that you put on top of this, for example, a sham, would be meaningless. So great result. And then this is what the loudness looks like. Again, we see a cumulative decrease in the loudness over the 6 weeks, which continues to decrease over the 6 to 12 washout period. And again, you can see, as with the TFI, that there was very little effect. There was some effect here with the TFI, possibly due to placebo. And there was some effect here, but we saw a significantly greater decrease in the loudness as well as in the TFI. So to remind you that a 6 dB decrease is a 50% reduction, and a 12 dB decrease is a 75% reduction. So we have up to a 75% reduction at the end of the 12 weeks. And you can go and look this all up in the JAMA publication if you want more details. So the second study replicated the pilot study, but with greatly improved longevity for loudness reduction and for TFI scores. The treatment effect remained even after active treatment ended, suggesting long-term neuroplasticity changes. The treatment effect was so pronounced that subjects did not return to baseline during the washout periods. So to summarize what I've shown you is that I've shown you a very small amount, as you can imagine, in 30 minutes of 2 decades of the systematic studies that were necessary. First, of the normal circuitry, showing long-term plasticity in that circuit in vivo, and then showing you that you have to have an animal model of tinnitus to know if the animal has tinnitus. And we need both behavioral evidence and physiological evidence, which I showed you in terms of the increased spontaneous rates and the increased spontaneous synchrony. Then I showed you that the DCN circuit changes with noise exposure, and that the DCN circuit changes in particular with tinnitus. And finally showed you how auditory somatosensory stimulation can reverse the tinnitus circuit in both animals and humans. And of course, these studies don't happen without people doing them. And so you can see, after over a couple of decades, there are a lot of people that worked in the lab and that contributed to these studies. But in particular, the people that are shown here are the people that were responsible for doing the studies that I showed you today. And so with that, I will take any questions.
Dr. Shore, that was absolutely terrific. Thank you so much. And we have a variety of questions. I think I'm going to start with some broad tinnitus, what causes them and so on type of questions. One of the questions was, is there a difference between drug-induced tinnitus versus noise-induced tinnitus versus other type of tinnitus?
Mm-hmm. Well, that is a really good question. And because our studies focused on, for the most part, on noise-induced tinnitus, but we also have done other studies, for example, with salicylate-induced tinnitus, where we were able to show the exact same processes that were happening in the brain. So I'm not going to say that for every kind of drug treatment that's going to be exactly the same, because it's very complicated. And it's going to be dependent on whether the drug is affecting the central nervous system directly or whether it's just affecting the cochlea. So I would say if it's just affecting the cochlea, we're going to have effects that are very similar to what we've been showing with noise exposure. But if the drug directly affects the cochlear nucleus neurons or the cortical neurons, then I think it's going to be hard to replicate the model exactly.
Another question had to do with how some tinnitus is helped by sound and some is made worse by sound. Your thoughts about the underlying mechanisms or differences between those two?
Well, the studies that I'm not an expert on sound stimulation, but the studies that use sound stimulation are often using it as a distraction. For example, as masking. Although there are some studies that claim that there are plasticity changes in the brain with sound stimulation. So our studies would go against that grain because, at least at the level of the cochlear nucleus, sound stimulation is not going to produce long-term plasticity. In terms of whether it makes the tinnitus worse, I think that there's going to have to, there has to be more research done to investigate that. I know it's true because it has affected my tinnitus sometimes. Sounds that don't appear to be damaging sounds can make tinnitus appear worse.
And sort of a generic question here as well, between tinnitus and hyperacusis. Are there similar mechanisms, or how do you think about those two different things? They're sometimes or often interrelated.
Right. Well, that just happens to be one of my pet peeves. And I think I made a strong point here about tinnitus being the presence of a sound in your head in the absence of a sound. And I said that for that reason, it's very important to report spontaneous rates. So in a way, it's a disorder of spontaneous rates in the nervous system. And that doesn't necessarily equate with a disorder of sound-evoked firing rates. So in our studies, for example, in the dorsal cochlear nucleus neurons, for the most part as a population, that population doesn't show an abnormal growth of firing rates with intensity, which is what you would expect to see in hyperacusis. You would see, instead of it growing in a linear fashion, it would grow in a expanded fashion, which would be what's indicating hyperacusis. So first of all, I don't think you can assume that the mechanisms are the same for tinnitus and hyperacusis. And I think that many studies use the term hyperactivity to discuss both, or increased central gain to discuss both. And I happen to disagree with that distinction because I think the kinds of, and I don't like to use the word hyperactivity because what does that mean? It means more activity. And so we know for tinnitus, there's more spontaneous activity. We know for hyperacusis, there's more sound-evoked activity. And these can be driven by different mechanisms, and even in our studies by different places. For example, we see that more in the ventral cochlear nucleus.
I have a two-part question, but it's part of the same linear question. We saw the dramatic differences between 4 weeks versus 6 weeks in terms of sustainability. And one of the members in the audience asked, can you see a build upon this work to achieve complete tinnitus suppression? Or do you think this is as far as bimodal stimulation can go? So I guess maybe talk about what you think is happening between 4 and 6 weeks, and can we make it even, obliterate the entire the stimulus?
Yeah, it's a great question. And I'll say that to be scientific, you need to look at trajectories. And so the trajectory of the pilot study showed a progressive decrease in loudness in TFI over 4 weeks. And because it went down like that, that was the reasoning for saying, okay, we'll do 6 weeks because it's going down, at 4 weeks. And what if we extend this? Is it going to go down and last for longer? And in fact, that's exactly what happened. So it was predicted based on the character of the decrement in the first study. So I think it's a good question, whoever asked that, is to say, if we do, 12 weeks, it can't really continue to go down because it's sort of leveled off. But is it going to keep going at 75% as we keep treating it, or is it going to go down to 90%? And the answer is, I don't know. And my prediction is that yes, that it would continue with repeated use, that the same trend would continue. Whether we don't know, we're going to have to know from real world studies, whether if you use it for 3 months, can you then stop using it? Or do you have to tune up, every 3 months? So these are going to have to be obtained from looking at real world data.
One of your of the bimodal stimulation, one of them is sort of a frequency specific auditory information based on their tinnitus. Is this treatment best for tonal tinnitus, or does it also work for broader tinnitus? Is one of the questions from our audience.
Yeah, that's also a good question. So, very few people's tinnitus is purely tonal. Even if it sounds tonal, it's never just going to be like a pure tone. And so we had quite a different range of, some people had narrower band tinnitus, some people had wider band tinnitus, and the results didn't seem to matter whether you had, very broadband tinnitus or more tonal tinnitus. The important point was that the treatment was the same as your tinnitus.
So I know one of the things that people talk about is how supratentorial inputs and perception of tinnitus and so on. Do you think this treatment that you talked about leads to central nervous system changes as well? Or are most of the changes occurring at the cochlear nucleus level? What are your thoughts about, in general, what's going on with the intervention and in terms of brainstem as well as the brain?
Right. So first of all, the cochlear nucleus is central. You mean more central. So this also brings me to another peeve point of mine, in that very often people who do research in the auditory cortex will talk about recording from the auditory cortex and then looking at the changes in auditory cortex function after they've done cochlear damage. So it's like the change in auditory nerve input leads to this change in cortical plasticity, as if there was nothing in between it. Well, there's a lot in between it. And so you might make an incorrect assumption, for example, in saying that if you record from cortex and you do cochlear damage, that there's going to be a reduced activity coming into the auditory cortex. But we know from the cochlear nucleus studies that that's not true. It's the opposite. So the input that comes from the cochlear nucleus is heightened, right? Because we just showed that. So you can't assume that now there's decreased input coming to the auditory cortex, because that increased input, that increased activity that I showed you is going to be transmitted to the inferior colliculus, to the thalamus, and to the cortex. So I think that it would be useful for investigators to think about what comes from the cochlear nucleus, and that's going to be the input to the higher centers, not the cochlea, because the cochlea only projects to the cochlear nucleus, not to the higher centers. So a long way of getting to actually answer your question is that, yes, I think that, and I know, in fact, because we've recorded simultaneously from cortex, and we show similar changes in the cortex, not exactly the same, but similar changes in cortex demonstrating spike timing dependent plasticity that coincide with what we're recording in the cochlear nucleus.
So this may be, you may have just answered the question I'm about to ask next from one of our audience members, which is, can you use your guinea pig model and determine some of the factors that predict resilience of, the maintenance of treatment outcome? And so you can take that question any way you want to. People are looking to see what will, sustain the treatment, and if there's predictors out there for it.
Yeah, so there's still a lot more work to be done that others have to do now. They can. There's a lot of work that still needs to be done. Following on from what I presented here, the question of resilience, I think, is very important in the way of looking at how the animals that don't get tinnitus are managing to resist getting tinnitus. So I think that that's an important question that many groups just don't do. you generate tinnitus, you just ignore the animals that don't get tinnitus. But I think those animals need to be studied as much as the animals that do get tinnitus. So one of the spike time independent curves that I showed you showed that the animals that didn't get tinnitus were much less plastic in this particular manner than the ones that did get tinnitus. So investigating what produces that less plasticity to the same kind of stimuli, I think would be a place to start.
Gotcha. And one of the questions, or several questions, speak to the bimodal stimulation. Can that be a generic treatment for any kind of tinnitus, or do you think there's particularly better candidates for it than other types of tinnitus? Or what are your thoughts about, in general, what predicts success after bimodal stimulation? Or do you think all comers can have success?
Well, I don't really like the use of the word bimodal stimulation, and prefer to use bisensory because that's specifically what we did. And bimodal just means two modes, so it could just be any two stimuli. So it's very specific, these two sensory stimuli at these particular intervals and orders that produce this plasticity. So that's first. And second, because we're activating the somatosensory inputs into that dorsal cochlear nucleus circuit, our initial thought was that this is going to be more successful with people who have a somatosensory component of their tinnitus. That is, they can modulate their tinnitus by some maneuver. And looking at various studies across the board, that turns out to be about 2/3 of people with tinnitus, regardless of what caused their tinnitus. So you can modulate your tinnitus if it was, induced by drugs, or if it was induced by noise, or genetics even. Somatosensory involvement of tinnitus is occurring pretty much wherever you have cochlear damage, because you get the somatosensory compensation. And plus, there are also other disorders like TMJ that are probably, they may need cochlear damage or not. That hasn't really been investigated. So yes, I would say that this covers a wide range of tinnitus etiologies.
So I'm letting the audience, by the way, the error about bimodal versus bisensory, that was mine, not our audience members, so I apologize. We're coming close to our end. I have two questions. One I really find intriguing. When somebody asked, if I have two different tinnitus, I have tinnitus both ears, they're different frequencies, will treating one ear have beneficial effect for treatment of the other ear?
Right, that's also a really good question. And I can only give you anecdotal anecdotal answer to that, is that in our pilot studies, we had a couple of people that received the treatment before we, just to get the treatment started. And one of them was an audiologist who did the treatment on herself. She had bilateral tinnitus, and she reported that her tinnitus went away or went down in both ears, even though we were only treating one ear. So I think that makes scientific sense, because there are pathways connecting the two cochlear nuclei, and they're going to influence each other. So I think, yes, I think whether they've got two different frequencies, I don't know. That would have to be investigated in a bit more detail.
So I think this will be the last question, and it's very broad, and please feel free to take it anywhere you like to go with it. What do you believe are the most significant unanswered questions in tinnitus research that will likely have an impact on current and future treatment options? But really, they can take that question any way you want and take us anywhere.
Well, I sort of think I've already answered that for you. I think that, one of the big questions is about resilience, of, why some people can have huge amounts of noise damage and even hearing loss and don't get tinnitus, and others who have a very small amount of cochlear damage can get a lot of tinnitus. So amount of cochlear damage doesn't necessarily predict amount of tinnitus or loudness of tinnitus. And so I think that is a big question that has to be investigated. I think that it would be nice if what we've shown in the cochlear nucleus were investigated systematically in higher nuclei. But I think the emphasis here is on the word systematic, as I've tried to show you here, in that just doing poking around here and there is less likely to lead to some result than doing systematic research, showing, first of all, what a normal system does, what a noise exposure system does, and then dividing that into animals that receive tinnitus and that don't receive tinnitus. Because if you just do noise exposure and you just look at, if we did that, for example, they would have canceled each other out, the animals with and without tinnitus. So we probably would have seen nothing. So it's essential to divide those groups into animals with and without tinnitus. And in the same, it's more difficult to do with people. But for basic research, I think those are very important.
Well, with that, I think we're going to bring this wonderful webinar to an end. Dr. Shore, that was just amazing, really awesome talk. Very clearly a large audience and a very engaged audience and terrific questions. So I want to thank all of you, our attendees, for coming to the webinar, and Dr. Shore for this very informative, cutting-edge presentation. We're so grateful to you, our community, for your support of our Emerging Research Grants Program. Remember that you can donate to our efforts to advance better treatments and cures for hearing and balance conditions by going to hhf.org/donate. Thank you, and please enjoy the rest of your day and a wonderful summer day here in New York City. Thank you very much.
Thank you, I know.